Thanks for coming, everyone. I hope that the Zoom people can hear us. It's a great pleasure to introduce Mathilde, who's coming to us all the way from Zurich, very far away. So Mathilde works in acoustic communication and bioacoustics. Before and during her PhD, she focused on marine mammals, worked on humpback whales in Madagascar and Cape fur seals in South Africa. In 2023, she received the FISIN postdoc fellowship and joined Martha Manser's group in Zurich. And then she also made a switch from marine to terrestrial systems and started working on meerkats. But today she will go back to her PhD work, and I'm very curious because I don't know that part.
Cool. Thank you very much, Vlad, for the introduction. Thank you, everybody, for coming here today and for being on Zoom. I'm very excited to share my work with you today. So as Vlad mentioned, this is my PhD work, but I just quickly wanted to introduce my current research in Zurich in Martha Manser's group. This is about meerkats, obviously, and I'm working in collaboration with Ari and Vlad on their amazing data set from the audio and color and GPS color data on the meerkats that we deploy in the Kalahari. And the aim of my research is to understand how the close call produced by meerkats during foraging help maintaining the cohesion of the group, but can also lead to the movement and the direction of the group during foraging. So this is a bit more about collective behavior, a bit more about the topic of this seminar usually, but I won't go into details on this study today because I have results that are a bit too preliminary, so maybe next time. But I really hope you will enjoy the still study, even if it's not really collective behavior. So yeah, let's go back to this, and I will talk to you about the acoustic communication network of the Cape fur seal. This was a collaborative work with different people from Paris-Saclay Institute of Neurosciences, and also people from Sea Search Research and Conservation in South Africa. Yeah, let's start with a quick introduction. And first, I wanted to give you a reminder of how we define animal communication. So animal communication is basically an exchange of information from one emitter to a receiver through a signal. And this transfer of information lead to a response of the receiver. It can be behavioral or physiological, but this response is an important part of the process. This signal can be of different nature depending on the modality, the sensory modality used by animals to communicate. This signal can be visual, acoustic, tactile, or chemical. And this exchange of information, this visual, signals, they have a major role in all social interactions for animals linked with their reproduction, foraging, anti-predator strategies. You all know about that. Today we will focus a little bit more on acoustic communication, so the exchange of information through a sound wave. And acoustic communication is the first modality used by birds and mammals to communicate. It has a lot of advantages because of the propagation properties of the signal first, because sound wave can propagate very fast and very far. This is very nice to allow communication among individuals. Acoustic communication is also reliable in the dark and can be easily localized compared to other types of signal, like olfactory cues, for example. And also acoustic signals have a great information encoding capacities. You can transfer a lot of different types of information with acoustic signals. You can give information about social and or ecological context. You can give information about you, your identity, your group, your species, your age, your sex, for example. This is what we call the static information. And you can also give information about your emotional state or your arousal state, for example. And this is what we call the dynamic information. So acoustic signals are very nice for all this reason and also very reliable in animal communication. But Of course, there's also some disadvantages to acoustic communication, as you can imagine. Noise is one of the main constraints because noise can limit the propagation of the signal. It can also filter the information that is transmitted. You can have some overlap going on, and you have also different physical phenomenon, like the constraint by physical barriers in the very dense environment, such as in forest, but also attenuation, refraction, a lot of phenomenon that I won't go into detail today, but just to keep in mind that it's not so easy to communicate via acoustic signals. So when we talk about this communication, we always think that there is one, not always, but mostly think that there is one emitter talking to one receiver. But in the wild, it's more complicated than this. You have several emitters, several receivers, and in some environment, it can complexify a lot the acoustic communication. And in particularly in colonial species, as you can imagine, the more individuals you have, the more complicated can be the communication among them. And there is a little bit of study about that in seabirds and especially in penguins. And on mammals, the only thing we know about communication in colonial species is on pinnipeds. So in this study, we wanted to know more a little bit on how colonial species can communicate in their environment in these big colonies, and also how they overcome this constraint. So that was the main goal of my PhD, and we once again went to the pinnipeds because they are very colonial mammals. And my study model was the Cape fur seal. So this is not a right species living in southern Africa, on the coast of Namibia and South Africa, where they form massive breeding colonies during the breeding season. And the Cape fur seal is one of the most colonial mammals in the world. The colonies can go up to 2010 individuals at the same place at the same moment, like here in Cape Cross in Namibia, here in central Namibia, where you have all these individuals gathering from early November to mid-January every year. I will show you a little video on how Cape Cross can look like. So as you can see, as you can hear, these guys, they make an extensive use of acoustic communication to allow their interaction during the breeding season. And this acts as a massive constraint for acoustic communication because of the high background noise and the high risk of confusion among individuals. And the second aspect that is very nice with this study is the structure, the social structure of the breeding colonies. So the colonies, they are formed in harems. So you have one adult male here with up to 30 females and their pup. The territorial male will hold the female in their territory to mate with them when they are ready to mate. And then you also have the presence of subadult males. So these guys, they are smaller. They are also younger, and they are not mature enough to get a territory. So they will roam in the colony and sneakily look for mating opportunities where they can, basically, and try to sometimes challenge the territorial males. For the mother and pup interaction, so the females give birth to one pup every year that they will feed for 9 to 11 months, so a very long lactation period. There is no allosuckling in this species, meaning that the pup can be fed by its mother only. It relies only on its mom to get food and to survive. And there is frequent mother-pup separation in this species because the females, 6 days after giving birth, they will go at sea for a few days and to get food themselves. And they will leave the pup alone in the colony. And when they come back, they have to reunite with the pup to be able to feed the pup. So all this structure and this like behavioral ecology of the species is kind of complex and is also very challenging in terms of pup rearing period. So this is a very nice study model to look at the acoustic communication during the breeding season. So in my PhD, I had several research objectives. Just to give you a very broad overview, the first part was about describing the vocal repertoire of the species, because we had absolutely no idea. Maybe not no idea because it's kind of similar to fur seal, but we had no study on their acoustic communication before. The second part was more about the information they can encode in their acoustic signals and how they can recognize each other through their vocalization in terms of mother-pup interaction, but also among territorial males. And because the study site I was working is highly exposed to tourism activities, I wanted to investigate a little bit the impact of anthropogenic noise, especially boat and car motor noise, on the behavior of these guys during the breeding season. So today I will only focus on a few aspects. I will present to you the vocal repertoire of the species and then show you how we investigate mother-pup vocal recognition and vocal recognition among males. Just as a very general method on how we collected data and perform experiments for these studies, I did work on several study sites during my PhD, but the main study site was Pelican Point in central Namibia. So Pelican Point is a 90,000 individual colony. It's located at the tip of this very long, sandy and flat peninsula here in Namibia, along a lagoon. We use a method, very simple method, to identify individuals, and especially pups. We use hair dye, blonde hair dye because they are black, and we kind of draw some numbers on their fur to be able to recognize them during the breeding season. As you can see here, it results in these marks. We had, for some studies, to track some births. So either we would do it directly by observing females giving birth, but we could also use the placenta release of the females, and especially the girls gathering to the placenta to eat it, to have like a good idea of when we had a birth. We could estimate the age of the pups based on their umbilical cord. The length and also the dryness of the umbilical cord would give us an idea of their age. And we, of course, perform some data collection, acoustic data with mic. As you can see, we are laying down in the colony because this guy, they really don't like when we stand up, and they can get very scared of that, and they can all go to the water. So I basically spend my whole PhD crawling in the sand to approach. But if you keep low and be very slow and nice, they would allow you to approach very close. And we also perform playback experiment. I will come back to this to present you some result. Okay, so let's start maybe with the first part, that is about vocal repertoire and identity encoding. So we describe different types of vocalization in that species. The first is a pup attraction call. It's a call produced by females to communicate with the pup. And I will broadcast to you 3 different calls from 3 different females. So this is how it sounds like, and you have also, in response, the female attraction call emitted by the pup to respond to its mom. And again, 3 different pups. Okay. We have different call types. The bark is emitted by adults, so male and female, and this is in male mostly for territory holding, so advertise your presence in your territory and keep your rivals away. And for females, it's more during agonistic interaction, when female would fight for this specific spot, because 2 females want to sit at this specific spot. They would produce bark and use this vocalization for agonistic interaction. This is how it sounds like. So it's very short vocalization produced in a sequence. And also we have other types of calls like the growls that are also produced during agonistic interaction, but this is more during a high arousal state when 2 individuals are just about to fight or to get into physical contacts. So yeah, that's a growl. And then we have describing another call type that is the long bark. It's basically produced by really a few males that would bring something a bit weird before and after a bark sequence. We have no idea why they do that and if it's really a signal or if it's a byproduct of their bark production. So this is probably not the most important call type to talk about. So we describe these call types by measuring different acoustic features in terms of frequency, duration, spectrum. And we did a discriminant analysis to see how this vocal repertoire would be spatially organized in a two-dimension plan. And this is what we get. So we have a very nice spread of the different call types in the acoustic space. They are mostly differentiated by their duration and also their average pitch. So this was a first result to show that their vocal repertoire is kind of limited, kind of small, with like 5 or 4 call types. But all the call types are very different in terms of acoustic features. And this is probably very helpful in a very colonial environment to have call types that are very different from each other and to be able to pick them from the background noise. So this is the acoustic partitioning of the Cape Fur seal vocal repertoire here. And then we wonder, do these individuals can encode their identity in their vocalization? So we asked this question in 3 call types, so the vocalization produced by female and pups, and also the barks from the territorial male. So we selected in our database different calls from different individuals, and we measured a number of acoustic features on their calls. And then we used a classification method, a random forest, to be able to calculate an index of vocal stereotyping. So this index will basically give us, are this vocalization individualized or not? And this index is calculated like this. You take the correct classification rate of your random forest. So basically, how good is your classification to discriminate between vocalization from different individuals? And you divide it by the chance which can be the correct classification rate obtained if the random forest would be completely done randomly, basically. So this is a nice index to have an idea of how individualized are the calls. But the only tricky thing with that is that this IVS is really dependent on the sample size, because depending on if you have 10 calls to discriminate or 400, your random forest will not perform as good if you have a lot of different vocalization. And also the chance will change quite a lot depending on the sample size. So it's kind of difficult to compare 2 studies that have different sample size with this index. So what we've done is that we calculated this index of vocal stereotypy over a large range of different sample size using a very simple bootstrap method. And we ended up with these curves that kind of show the IVS range depending on the sample size you take for the species. So you have 2 curves because it's 2 different colonies, but just take it as a Cape Fur seal average index of vocal stereotypy. And then you can bring different studies from past research and put the IVS of other species of pinnipeds. And for one sample size, like here, like a sample size of 8, you can compare between different species how individualized are the vocalization. So this is for the female's vocalization. And you can see that for most of the sample size, the Cape Fur seal is approximately on the top of the IVS values in the pinnipeds. We have the same result with the pup vocalization. Most of the time, they are more individualized than in other pinniped species. And for the barks of the territorial males, we don't have a lot of studies, so it's a bit more tricky to interpret. But basically, what we've shown with this first study is the acoustic partitioning of Cape Fur seal vocal repertoire and how it can be advantageous in a very dense and noisy environment, and also a high degree of individuality in their vocalization. And I didn't present to you, but this identity is encoded in different acoustic features that are mostly the duration and the fundamental frequency, which we call F0, and the frequency modulation of the call. So basically, we have calls that are very different from one individual to another. The next question was, are these calls involved in the vocal recognition? Can they extract this information about the identity of the emitter, and can they react according to this identity? So that's the second part about vocal recognition in mother, pup, and pair. So first part of this study is, do we have a vocal recognition between mother and pup? And to do that, to test this, we use playback experiment in which we broadcasted different series to females and pups. So basically, when we tested a female, we would broadcast 2 different series, her own pup's call and a stranger pup's call, and see how she reacts differently to these 2 series. We did the exact same for the pups. The order of the series was randomized, and the playback track for each series was something like this: 6 different calls from the same individual, either own pup or stranger, separated with a 2-second interval, which is approximately the natural call rate of this species, and for a duration of 15 seconds. And then after playing back this track and a video recording the response of the individual, we evaluated their response by looking at how they look toward the speaker during the experiment, how they change their posture, how they call in response to the playback, and also for the female, whether they check their pup or not. Because some female can check the speaker and some female can check the pup. So we made this variable especially for females. And with these behavioral variables, we calculated the PC score resulting from a very simple PCA to get a composite score that gives us the strength of the behavioral response of one individual for the 2 series. I will show you here 2 different series. So this is a test on a female. Sorry. So that's the female I want to test. That's the speaker, and this is her pup. Is sleeping on her side here. And the first series is a non-filial series. So that's from a stranger pup. And just to help you, I will mention every time we have a call from the playback. So we have 6 different calls. Yeah, that's the playback. 2, 3, 4, 5, and 6. So as you can see, there is no obvious response of the female. She is not passionate by the playback at all. But now if you test her with the filial series, so her own pup vocalization, the pup is still sleeping here, the female is laying down but she's not sleeping, she's completely awake, and we would broadcast the vocalization from her pup. 1, 2, 3, 4, 5, and 6. So very good recognition. She called back in response to the playback. She changed posture. And as you could see, she was very reacting to the speaker, but not to the pup that is sleeping right here. So she really focused on the sound source, but not what could produce this sound, basically. So here I will show you the result for the whole data set. So here is the PC score on the y-axis. The PC score is reversed because we have strong behavioral responses on the negative. So don't mind the order of the axis. Just keep in mind that the strong behavioral response are on top. And for females, you can see that we had a significant stronger response for mother to their own pup's vocalization compared to the stranger pups, as you could see in the video. And we had the exact same result for the pups. They responded more to their mother's vocalization. So this shows that we do have a vocal recognition in this species between mother and pup, and that this recognition is mutual because both female and pup can recognize each other. It's not always the case. We have pinniped species in which the pup cannot recognize its mom, for example. In this species, both of them are very good and can rely on acoustic signals. The question we ask after that is, okay, they can recognize each other, but when do this recognition establish? We know that the female first departure is 6 days after parturition. We expect this recognition to be established during these 6 first days, because otherwise she wouldn't be able to recognize the pup when she's back. But we had no idea whether they could do that very quickly or if it needs a bit of time. So we used the exact same playback experiment setup, but we did test each individual several times at different time after the birth. So here are the categories we did between 2 and 4 hours after the birth, 4 to 6, etc. These are for the females and these were for the pups. In terms of data collection, it was a bit more challenging because we had to do everything in 2 hours after the birth. So we spotted the birth, recorded the vocalization, marked the pup, create the playback track, and then perform the first playback 2 hours after the birth. And what we obtain is kind of very cool. We have the same plot here with the strong behavioral response on top. And here for the female, you can see that as early as 2 to 4 hours, they already responded more to their filial series compared to the non-filial series. So 2 to 4 hours after the pup birth, they were already able to recognize the vocalization of their young. And for the pups, it was a little bit different, but not so much. They responded the same at 2 to 4 hours, but after 4 to 6 hours, they were already able to recognize their mom vocalization. To be honest, we expected them to do that in a few days, so we planned to do the experiment over several days. We had to do it after several hours, and then we stopped because they were so good at recognizing. This was kind of impressive. And yeah, this study showed that, oh no, sorry, I'm forgetting an important part. The next question we asked with that was, okay, they can recognize each other after a few hours, but does the vocal activity of the female impact the recognition of the pup and vice versa? Because if you are highly exposed to the vocalization, maybe you would learn them quicker. And also because we saw a lot of differences between individuals on how vocal they were after the birth. Some females were highly vocal, producing a lot of calls. Some others were exhausted by the birth and would just go to sleep. So we wanted to know whether the pup can be better at recognizing if the female was highly vocal after the birth. So these are the results for the pups. So this is basically the response between the filial and non-filial series for the first playback, performed at 2 to 4 hours, depending on whether the pup was very vocal or not during the first 2 hours. And what we could see is that there is absolutely no relationship between the two. And even if the pup was poorly vocal the first 2 hours, the female were very good at recognizing them. And we had the exact same result for the pups, no relationship between the vocal activity of the female and their ability to recognize. And as I told you, 2 to 4 hours, we had no significant differences between the two series, but we still had a little bit of individuals that were able to recognize their female because they responded more to the mother series compared to the non-mother series. So this is not significant at 2 to 4 hours because it's not the majority of the pups that responded, but still we had some that could recognize. And what is interesting here is that some of the pups here that were able to recognize had females, mother that were very poorly vocal during the first 2 hours. Some of them had call rate less than 3 calls every 10 minutes. So they were poorly exposed to their mother vocalization during the first 2 hours. And here I will show you the video for the pup. So this is a pup that we tested 3 hours after birth. This is its mother. She's completely exhausted by the birth, so she's sleeping. And he's kind of trying to suckle, but that's the back, so he's on the wrong side, but he's trying to find the nipples to suckle. And I will show you the first series we did with him, with the non-mother calls. So that's the first, second, third. So the pup is not really interested in the playback. He's still trying to find the nipples and like moving around his mom, but not really interested in the calls. But then if we test the pup with the mother series, we have the same organization with the loudspeaker on the right here. So the first call was a bit overlapped with something else, but then after the second call, he raised the head and is calling. Looking at the speaker and calling, I think he produced 7 calls in response to the playback in like 20 seconds. So highly motivated to respond. And this pup was pup 29, and pup 29, his mom called only once in 2 hours. As you could see, she was completely exhausted, and we had only one vocalization to put in the playback track because we could manage to record one vocalization in 2 hours. So even if he could hear his mom only once, he was still able to do a very good response to the playback series. So this result lead us to the hypothesis that we could maybe have in this species an in utero vocal imprinting, because the females, when they are pregnant, they are taking care of the pup from last year. They are always lactating and pregnant. So it means that during the 11 months of pregnancy, the pup is growing with the female calling for his sibling from the last year. So we think, we didn't test that, and it's very difficult to test, but that these fetuses can be imprinted on their mother voice in utero. And this is what this result suggested. So yeah, we showed mutual vocal recognition in this species with a synchronous onset, with both the female and the pup being able to recognize each other very quickly after birth. And this is the fastest establishment for any mammalian species. Just for you to give you an idea, the fastest establishment of mother-young recognition was previously shown in sheep, and it was 12 hours for the females and 24 hours for the young. And in humans, I think it's half of the females, half of the mother that can recognize their baby cries after 1 month. So they are really good, way better than we are, but also they obviously have more constraint than us for vocal recognition. But these results were quite impressive in terms of what we know on the mammalian species. Yeah, we showed that there is an asymmetrical vocal imprinting period because I didn't explain that, but the pups are way more vocal during the first 2 hours. As soon as they are born, they keep vocalizing. So the females are highly exposed to their pup's vocalization during the first 2 hours, which is not always the case on the female side. But we have no effect of the vocal activity on their ability to recognize early or not. And yeah, as I mentioned, we suggest an in utero vocal imprinting in that species. Okay, let's go with the third part, and now we'll talk about vocal recognition among territorial males. So this is how the colony looks like in terms of structure. One territorial bull, we call them bull, they are older than 13 years old to keep a territory. Younger than this, they are not mature enough, and we call them subadults. So one territorial bull with mother and pups. The pups are not the pups from this bull, right? They are from the last year, so they are not their offspring, but they just keep the female to mate with them, basically, and they absolutely don't care about the pups. And then around you have some adult males that are younger. The adult males, they are mature enough to have a territory, but they just don't have a territory. They didn't get the opportunity. And then you have the subadult males. Those guys won't have a territory at all. They are not mature enough and strong enough to challenge the big adult males. And then, as I told you, the males, they produce barks for territory display. So they basically spend their whole day producing barks in their territory. During the breeding period, they don't go at sea at all. They fast for 2 or 3 months. So this is a very challenging time for them. They keep being challenged by other individuals. They don't get energy. They don't get food. So they spend their time barking and showing other individuals that they have a territory, don't mess with me, basically. So in this study, we wonder whether the territorial bulls could recognize each other. Can they recognize their neighbor? Here, I know that I have a neighbor with his territory right next to mine. Can I recognize his voice and just make sure that we won't fight every time we hear each other barks, because it's very energy consuming. So it would be more advantageous for them to know about their neighbors and be aware of them being there, but not fight with them every time they hear them. So that was the goal of that study, showing if there is a neighbor-stranger recognition in the territorial males. And for that, we use playback experiment, again, where we have one focal individual, that's the territorial male we want to test. This guy has a neighbor on this right side, for example. And the first series of the playback would be broadcasting the neighbor's bark from the right side, so from his territory, on the congruent side. Then we would do that from the left side, broadcasting the neighbor of the right, but from the left side. And we would also broadcast a stranger territorial males that is far away in the colony, also on this left side, to compare the different responses. The behavioral response was assessed by the look toward the speaker, the latency they changed to the latency for posture change, and also whether they produce barks in response to the playback. We also obtained the PC score to have the score of the behavioral response. And as before, strong behavioral responses on top of the plot. And here you can observe we have this result. So there is different way of looking at this result. First, you can see that they responded more to their neighbor on the wrong side compared to a stranger. So this means that we do have vocal recognition between these guys, otherwise they would respond the same to their neighbor and to the stranger. So we know that they can recognize each other based on the barks. Then the second result is they responded more to their neighbor when the neighbor is broadcasted on the wrong side compared to the right side. So this showed that the focal males find their neighbors more threatening when they think that the neighbor left his territory and moved away from his territory. So they kind of have an idea of what neighbors I have, what are their barks, acoustic feature. I know I can recognize them, but I also know where they are located respectively to my territory. And if you move them and you pretend that one individual, one neighbor change location, then they would get very nervous about that. And the last result is that they responded more to the neighbor on the wrong side than a stranger. So it means that the focal males find their neighbor outside their territory more threatening than a stranger male. This we didn't really expect. We saw that either the neighbor outside his territory would be threatening, but a stranger male can be also very threatening. So we expected the response to be the same here, basically. So in this study, we showed that there is individual vocal recognition among territorial males in that species. They have what we call a mental map of their neighbor's territory position that is associated with their recognition of their barks. And also, we show that the neighbors outside the territory are considered threatening. And this, we interpreted this result by thinking that the stranger males, when you have a stranger coming to your territory, you don't know if it's a subadult, you don't know if it's an adult, you don't really know how challenging this guy can be. But if your neighbor that you know he's a big guy, he has a territory, and he moved away from his territory, it can maybe to challenge your territory and take your females. So we think that maybe for them it's more advantageous to be very careful with their neighbor, because they know that these guys have the maturity and the strength to compete with other males and take their females, basically. So yeah, with that, I would like to thank you all my collaborators, the students who worked for that project, and all the funding sources. And I also would like to thank you for your attention. I'm very happy to answer any question if you have. Thank you.
think we have time for questions.
Thank you for the talk.
No.
I was wondering, they have pups every year.
Mm-hmm.
So what happens in the second year? And I mean, the next year, does the pup change the sound? Does the mother get a new sound, or does she have the same one? And does the now 1-year-old pup respond to that? What kind of confusion do you see from 1 year to the next?
Okay, so as soon as they give birth to a pup, the pup from the previous year will be kindly asked to leave, right? They can't really feed two pups at the same time. It's very challenging for them. So some pups, like the yearling, we call them yearling when they are 1 year old, they try to stay with their mom and get some more milk. But normally they are weaned at this age, and the females will stop taking care of them and just chase them away so they can feed on their own. So they are very rarely exposed to two pups at the same time. And also the pups from the next year doesn't always have the same acoustic features in the vocalization, because these acoustic features depend on a lot of things, their body size, their anatomy, and things like this. And because the male, the father of the pup is not the same between years, there is no reason that the vocalization would be the same. So she basically has to learn a new vocalization pattern every year. And what is interesting as well is that the pups' vocalization, they change over time, and depending on whether they are 4 days or 2 weeks, the vocalization will improve a little bit. They will also increase their body size. So the female, they constantly have to learn a new version of their pup voice, basically. And they are very good at doing this. And if you test a female that has a pup that is 4 months old, and you test this female with the vocalization you recorded 3 months before, she would remember the version of the pup's vocalization. So they really keep in mind this vocalization evolution over time. So yeah, they constantly have to learn, basically, what's going on.
And the female does not change the vocalization?
No, the female are very constant. Yeah. It can change a little bit if they are very, if the emotional state is changed, basically. If they are very nervous, sometimes they take a few hours to find the pup in the colony, and after a few hours they can get very nervous and go a bit high pitch and try to find the pup. And as we could do, the voice can change a bit if you are very motivated or very nervous. But usually it's very stable, yeah.
Also, very, very good talk. Thank you.
Thank you.
I was wondering about this last part that you were talking about. So how stable are the territories, and how often do you think the males, if they're not very stable, the other males are changing positions, whether they have to constantly update this mental map of the location of other individuals?
Yeah. It's very difficult to assess how stable are the territory because we don't manage to mark the territorial males. They really hate to be touched, so we try to mark them with different ways, but we never manage to do that. So we have the feeling it's changing a little bit over the season, also because for them it's very difficult to keep a territory for a few weeks. I mean, they don't feed, they don't get food, and they spend their time being alert and vigilant. So I don't think they would spend the whole breeding season having a territory. And also not the structure, but the shape of the colony will change over time. So in Namibia here in the morning it can be very cold, but then after 9 or 10 it gets very hot, and all the individuals kind of fight to get close to the water to thermoregulate. They like to sit in the very shallow water and hold their territory from this place. So the movement of the territory is constantly moving from the morning to the afternoon. So the territory are a bit changing. So I really think they have to update very often what's going on around them, and also because there is absolutely no physical cues of the territory. It's just individuals being close to each other, and one female, if she is in between two territories, they don't really know if it's mine or it's yours. So they kind of try to keep their territory border stable, but with all this dynamics of colony moving and individuals moving, and so yeah, and also the females want to move. The females, they are not attached to one harem. They don't really care. So sometimes they just want to go to the water, and the male try to hold the female. No, you don't go. You stay with me in the territory, and they don't go. So yeah, it's very dynamic.
I was wondering, how long does the female leave her pup when she goes for food, and how much does the song of the pup change in this time? So how robust must this be to a change in the song for her to recognize?
So at the beginning, it's a few hours. They just go and try to get some food and then come back very rapidly. As the pup grow, it can be up to 4 or 5 days. They don't have to be too long, otherwise the pup won't survive. So yeah, it can be a few days, and I don't think in a few days the vocalization will change that much. So yeah, I don't think they would experience any problems in that sense. Yeah.
Thank you for the really nice talk. I was wondering, I know the logistics are very complicated and I've never done fieldwork, but were you able to follow females like over the course of having 2 offspring? Like, no.
No.
Okay. Yeah, I mean, I figured, because I was just wondering, because that would be very interesting to then be able to look at like how much did that female speak or vocalize to its previous offspring, and if this then actually is correlated with, to look at the imprinting. But
absolutely, that would be amazing. But unfortunately, we were allowed to mark them temporarily. Yeah. So the females were not marked. We couldn't. They also don't like to be touched and to be marked, so we didn't bother them that much. And we just marked the pups, and it was just for the breeding season because they molt and then the dye would disappear. We would have loved to mark them with flipper tags that we could see and spot time after day, years after years. But yeah, no, we couldn't do that. And also I would have loved to test females, very young females for their first birth, and see whether the pup is very good at recognizing or not, because it has obviously not been imprinted during the pregnancy. But this we absolutely have no idea. So.
I just wanted to go back to the what you said. Like there is a change in the vocalization of the pup as it grows, and and so you said the mother constantly learn that change. And do you think it is like she's learning every time like those changes, or maybe there is like one constant feature that is not changing for the pup's calf, and that's what the mother kept in her mind, and that's how she's able to know even if there is some changes? Yeah,
I don't think there is only one feature that is stable, because a lot of changes are happening during the maturity of the pup. So it's about frequency modulation. When they are very young, they do that bleating that you could hear like, and then after a few months they stop doing this, so they can't rely on this bleating anymore. So I think it's the overall call that's changed in terms of frequency and spectrum. But I think that's very slow process learning. Time after time, they just get used to, as we do with, teenagers that have the voice changing over time. You obviously learn it because you are exposed to that very often. So yeah.
Thanks a lot for your talk. When you were doing the playback experiments, did you try also to quantify the similarity between the stranger pup and the...
Yes. Yeah, this is something I didn't mention, but we looked at... So we first did the playback with changing the stranger completely randomly. From our database, we would change whatever stranger pup we could find. And then I realized in some playbacks that we would broadcast a stranger that is not so similar or not so different from the actual playback series. So I quantified this by looking at the acoustic similarity between the 2 series. And I had... I think the more similar was 0.5. Like it's just a correlation of spectrogram basically. And if you have a 50% correlation in the spectrogram, they are still able to recognize very, very well, and they are not confused by the stranger. So I really think they rely on, they have a bunch of acoustic features they can rely on, and even if some sound can be a bit similar to them, it's still different in their mind. And also because this is very dense colonies, you have pups everywhere calling all the time. So they really have to be sharp in their recognition. Otherwise, they would be confused with other pups all the time. So yeah.
Thank you very much, and thanks for joining. Thanks for questions. If you want to chat with Mathilde a bit more, we're going to go and grab lunch in the Mensa, so just stay around and join us. Thank you.
Cool, thank you very much.